So hello, good evening everyone. Thank you for joining us. My name is Kim Aiken from the Faculty of Science and I'm filling in for Udi, who is our Alumni Engagement Manager. She's currently on maternity leave. So let me start by acknowledging the people of the Kulin Nations on whose land we, the panel members, are gathered on tonight. And I pay my respects to their elders past, present and emerging. I'd like to extend a warm welcome to our alumni joining us this evening from Australia and overseas, and also from within the different time zones within those areas. And a big welcome to our other members of the science community as well. Tonight is part of the Monash Science Alumni webinar series, where we wanted a platform to be able to share with you the latest research from Monash Science, but also to assist you with ongoing learning as well. The format of tonight's event will be a 30-minute presentation given by Professor Phil Andrews and Associate Professor Rico Tabor, with a 15 to 20-minute Q&A session at the end. We'll be taking questions from the Q&A function at the bottom of your screen. So as we go along the different presentations, please feel free to submit your questions through this channel. We won't be using the chat function tonight, but yeah, as I said, please feel free to enter your questions through the Q&A function. It's now my pleasure to invite the wonderful Professor Andrea Robinson, who is also the Deputy Head of our School of Chemistry, to commence this webinar. So Andrea will provide an overview of tonight's topic, introduce you to the panel members, and also facilitate the Q&A session at the end of the presentation. So I'll hand it over to you, Andrea. Thank you.
Okay, thank you very much, Kim. A very warm welcome to everybody on tonight's Zoom call. We're really delighted to have you with us tonight. It's my pleasure to introduce tonight's Monash Science webinar speakers, Professor Phil Andrews and Associate Professor Rico Tabor, and their presentation, Rise and Demise of the Microbes. Humans purposely and inadvertently introduce antimicrobial chemicals into their environment through the use of disinfectants, antiseptics, antibiotics, and everyday commodity items like detergents and toothpaste. So chemists have played a very long-standing and critical role in maintaining global health against invading microbes, and will continue to provide new solutions to combat multi-drug resistant microbial infection. Our first speaker tonight is Phil Andrews, who joined Monash in 1995 and is the current head of the School of Chemistry. Throughout his career, he has been the recipient of a Humboldt Fellowship, a Royal Society Fellowship, and an Australian Research Council QE2 Fellowship. And so I'm making him feel very uncomfortable. You can see him shifting in the background in there. So he is known internationally for his work on bioactive metal compounds, main group chemistry, and antimicrobial materials. Our second speaker tonight is Associate Professor Rico Tabor, and he completed his PhD in Bristol and joined us in 2012. We were absolutely delighted to recruit him into the School of Chemistry at Monash University, and he is an extremely popular undergraduate supervisor and lecturer and postgraduate supervisor as well. Rico is extremely passionate about studying surface and colloidal forces and their application within complex multi-component systems. His work perfectly interfaces, if you'll pardon the pun, with the organometallic chemistry of Professor Phil Andrews, and this is the reason why we've badged them together tonight. So it's my great pleasure now to hand over tonight's webinar to both Phil and Rico. Thanks, guys.
Thanks, Andrea. I guess I'll make a start. Welcome everyone to this webinar. The first part, I'm going to give the presentation, and obviously, as you can see from the title, it's about the rise and demise of microbes. I would have to think that there's been much more rising rather than demising of microbes, but at this point in history, I think we're at a point where we really need to consider the positive effect that microbes have, but also as we know over the last 2 years, the very negative effect that a microbe can have on the way we live and how we live. And so I'm not going to really talk about COVID. I think Rico is going to talk about COVID. I'm going to steer away from viruses, and really what I'm going to do is talk about the threat of antimicrobial resistance, because while the problem of COVID has been very deep and will be ongoing, in the background, we still have this almost cataclysmic problem of the rise of drug-resistant bacteria, which is ongoing and happening all the time. The first slide I want to show you is something which is happening at Monash and is inaugurated over the past couple of years, and that is the Monash Centre to Impact AMR. For those who don't know, AMR means antimicrobial resistance. And this has brought together researchers and faculties as far apart as social scientists into IT, into science, and of course in microbiology and other areas of biology and medicine itself, bringing these people together to talk about how we can overcome it. So if you want to learn much more about what's happening with AMR, there's many resources around the place, but I think the AMR Centre website is a really good place to start. So let me put it like this: back in 2014, and I'll give you a whistle-stop tour of AMR. There's a lot of information out there about it already. 2014, the World Health Organization issued a report. It had a look across the world to see what the impact was of drug-resistant bacteria and other forms of microbes. And here they are, and they said that there's a very real possibility for the first time since the introduction of penicillin back in 1928, when we looked at the first generation of antibiotics that were discovered largely by accident. We've had a very good run in terms of being able to treat infections very well. And we forget what it was like prior to that to actually have an infection, where if it was particularly serious, it would lead to death. But other than that, you could have amputation, serious lung infections, chronic disease for many years, and children contracting a disease could easily lead to infant mortality. And that's a real situation that we could be facing in the future. We have kind of forgotten, our generation, how serious that can actually be. So we have some problems, I think, in that we are going to face a future where minor injuries can possibly kill. So obviously we have to continue the fight to make sure that doesn't happen. So here is the main problem, and I'll do it in a couple of slides. So if you've been back 2011, so over a decade ago in Nature, they talked about the perfect storm. What was the perfect storm? The perfect storm was the increase in drug-resistant bacteria, which were being found and acknowledged and sequenced across the globe at the same time as they were finding that antibiotic resistance was on the rise, and that some of our frontline drugs were becoming ineffective in treating infections. The pharma industry, the people who had been driving the development of antibiotics, were getting out of the field. So you can see from that slide, in 1990, there was about 18 major pharmaceutical companies were investing in the development of antibiotic pipeline. By 2010, that was down to 4. And it still remains as low as that, despite the fact that the United Nations, World Health Organization, and many governments have now put in action plans to make sure there are One Health approaches to managing antibiotic resistance, but also maintaining that research and development pipeline. I'll talk a little about that in a second. So that's the perfect storm that we have. What are the causes of antibiotic resistance? Well, we know about the overprescription of antibiotics, and everybody talks about being given antibiotics when you've got a viral infection, which could be a simple respiratory infection. We know about patients not finishing their treatment and throwing their antibiotics away when suddenly they feel perfect and there's still enough in the system to mutate, and the antibiotic has killed off the weak ones, the strong ones live on and can mutate and become drug resistant. It's also used in agriculture and aquaculture, poor infection control in hospitals, and a general lack of hygiene. We've become perfect in the last 2 years of hygiene control. And I think, imagine if it kept that way. I don't think I've had a cold for the last 2 years. It's been amazing. And if it wasn't for hay fever, I think it would be a paradise at the moment. And the last one, of course, is the lack of new antibiotics that have been developed. That's a severe risk. What I'm going to talk about eventually is how we can challenge the problem that we have and the strategies that we need to do it. So why is Big Pharma getting out of developing antibiotics? This chart here, I think, gives you a clue. If we look down here and we look at the new generations of antibiotics that were being brought through, recognized by the FDA in the US, were being released onto the market, as soon as they were being released, within 4 to 6 months, resistance was developing. And that's a major problem. You invest billions of dollars in the development, the regulation, getting through the regulatory control, the experiments, doing the phase trials, getting the clinical trial cohorts together, making sure you understand the efficacy of the drug, and you release it onto the market within 4 months. Essentially, microbes have developed a resistance to it, and therefore there's no long-term efficacy. As whole classes of antibacterials, antibiotics were becoming ineffective. And so rather than continue to invest in a non-profitable exercise, these companies were pulling out. And that's the role of government and big health organizations, I think, now to start to challenge that process. So that's the reason why. The pipeline is not particularly good. If we look at what was happening in 2020, we have essentially got about 20 new antibiotics which have came through into phase 3 trialing, which is just short of being capable of being released into the market. Only 3 of those 20 compounds belong to a new class of antibiotic. And that means what's been happening is that traditional classes of antibiotics, which I'll show you in a minute, have been redeveloped, released, modified, and very quickly become ineffective. And that's a problem. If we're not developing new classes of antibiotics, then we're not going to challenge the evolutionary drive that the microbes themselves have to generate that drug resistance. And so we might need a different approach to that in some of that. This is from the Australian First National Antimicrobial Resistance Strategy. We can talk about the impact in general terms. So the OECD, as you can say, is that every year about 290 people are dying from drug-resistant infections. By 2050, when it's estimated that more people will die from infections than die from cancer, then that's going to be up to about 10,500 per year for drug resistant. And it's not only a health cost, of course, a personal cost, a human cost. It is, of course, an economic cost, and to the health system to try and manage the rise in infections is going to be significant. In fact, we're talking billions and billions of dollars across the world just to try and manage that situation. So something has to happen. So the World Health Organization told us what the priority pathogens were in 2016. And you can see that most of these, in fact, all of them are essentially drug resistant. Carbapenem resistant Helicobacter, Pseudomonas aeruginosa, which is a wound infection bacteria. 3 bugs that we look at on a regular basis, the enterococci, VRE, Staphylococcus, so that's MRSA, and Helicobacter. Helicobacter I'll talk about in a second, simply because it's been of interest in the way that we have developed our research around bismuth chemistry. So we know what the critical pathogens are. We just need new strategies and new compounds on top of all the hygiene and healthcare measures that have to be in place to manage this situation. So one of the major areas that we have a problem, of course, ironically, is in healthcare situations, so in clinics and healthcare. And we've realized that because of the impact of COVID, something that was generally hidden for a long time, has now become apparent that some of the most vulnerable places for cross-infection or for patients to get infection is actually in hospital. And you can see from the joke here. So there's a need to generate what we call clean surfaces. So those would be antimicrobial surfaces which inhibit biofilm formation or don't allow planktonic bacterial cells to settle and to be passed on simply by people touching. We need an environment within hospital settings and within surgical settings, even within GP settings, where when you go in there, you're not expected to actually catch an infection that could mean that you have a long-term health crisis associated with that. So there's a whole engineering, materials, chemistry, biology, medicine approach which needs to be used for this. So with this slide, I know that probably not everybody's a chemist that's on here and probably looks aghast when I put up chemical structures. But these are essentially the basic structures of most of our antibiotics at the moment. Most of you maybe recognize some of the classes of antibiotics, in particular fluoroquinolones, which now have gone through into their third generation, because the earlier generations are simply no use anymore. We've got beta-lactams, where you have lactamases, metallolactamases, which have evolved within bacteria, which is simply render a lot of these penicillin-type molecules now completely ineffective. And so the problem is when you start to think your strategy is simply one of regenerating new modifications of existing classes of antibiotics, the problem is it becomes really difficult to outrun nature. Microbes, bacteria have proven themselves enormously effective at using these environments, at using our environments, and using evolution simply to overcome the strategies that we have used to take them on. The thing I want to point out about these compounds is they're simply molecular organic molecules, so traditionally derived from nature or synthesized in a laboratory and then scaled up and put on through regulatory process and put onto the market. So they would have been developed in terms of what targets they would touch, how they would transport through the body, the life that they have in the body, how they're eliminated for the body, and the impact they have on the bacterial cells. And the bacterial cell, it has to be selective for over what we would consider to the background mammalian cell as well. So it has to be selective in what it does. And so it's taken on those bacteria. What I want to do is talk about something a little bit different, because my research is essentially using metals. And when we move from wholly organic or natural molecules, which are organic molecules, into metal-based compounds, then we're dealing with a whole different structure in the way that compounds are built. And we're going to use the strategy around how you build metal compounds and complexes to try and have a new way of combating the antibiotic resistant or antimicrobial resistant, which is developed. So new targeting strategies, which are actually multi-targeting strategies rather than looking at one single aspect of how you disrupt the cellular processes of a bacteria. So we've known for history has always dealt with this, right? So there's always been metals, silver, mercury, silver and copper were discovered 4000 BC. We know they've got antibacterial activity. And we look across the whole periodic table, we can see there's an array of metals that we can use that actually will allow us to disrupt the function of bacterial cells, and they can do it selectively, and that we can use those to try and develop new classes of the compounds. So we have generally in this area here, we have the essential metals like zinc, which exists in 300, 400 metalloenzymes in the body. So it's essential. You can't ignore iron, it's completely essential. And then you've got trace metals like copper as well. And you'll find the other metals involved in various enzymes throughout the body. You've then got other metals which are considered toxic: antimony, arsenic, mercury, who traditionally have been used for thousands of years in medicinal practice. Our interest has largely been around this element here, which is called bismuth. As you can see, it's in that heavy metal zone, and you often think that's going to be a bit tricky. Mercury in very small amounts can be lethal. Antimony can be problematic. You don't want to be drinking and swallowing too much lead or breathing it in, and therefore how does a metal which is surrounded by these highly toxic heavy metals actually make a difference? So these are some of the, I think Andrea in the blurb that went out for this webinar talked about mercurochrome. So I thought about putting mercurochrome in. So up until the 50s, mercurochrome was very common. Children used to develop Pink's disease because they would get mercury intoxication. That was well known. But today we've got a few metal complexes that is really well used. This is cisplatin, which is one of the most, if not the most common anti-cancer drug in the world, and it's predicated on that platinum center. There's a few others which have been developed. There's a few other drugs which have gone through to phase 3 trial, and this is gold, antimony. This one is used for the treatment of leishmaniasis, which is a parasitical infection that we also research. You've got ruthenium anti-cancer-based compounds. You've got anti-malarial iron-based compounds, and vanadium as anti-diabetic compounds, so insulin mimics. So it's in there, it's in the arsenal of compounds that we're trying to develop. The one I've been talking and interested in is bismuth. So bismuth, for some reason, and I could talk for hours on this, I'll try not to, is that bismuth is a heavy metal that has got incredibly low toxicity in humans, but is a very effective antimicrobial metal. We need to understand why that's the case. So in Nature Chemistry back in 2010, Ram Mohan wrote about green bismuth, so the environmentally friendly heavy metal. Now, you can't simply say that it's completely non-toxic. So later on, there was a more balanced approach where we talked about the medical use of bismuth, two sides to the coin. So it really depends what the structure of the compound is. Interestingly, Hong Zhe Sun, who's at the University of Hong Kong, he showed only last year, or 2020, 2019, that co-administering bismuth could knock out the metallo-beta-lactamase that was destroying those sulfonamide antibiotics. So co-administration of a bismuth compound with an adjuvant was enough to reactivate and essentially reinstall the activity of a class of drugs which is essentially fallen out of practice in those sulfonamides. And so that was a great breakthrough. We knew metals were going to do something. If you want to do some further reading, you can see at the bottom, we just wrote a review this year with a few colleagues, which we've released on medicinal bismuth. I won't bore you with that. This is Pepto-Bismol. I don't know how many of you would know Pepto-Bismol. If you go to the US, for example, you can buy it in any corner store. You can open it and you can drink it. And you can drink about 250 milliliters of it. The worst probably that's going to happen is your stools become black, your tongue becomes black. But it's used to treat traveler's diarrhea. And also now it's used to treat Helicobacter infection. And we looked at the structure of this back in 2006, and we showed that these were big bismuth oxide type structures that were contained within that. But there aren't very heavy metals you would go into a corner store and buy a bottle of and just drink it to treat your gastrointestinal problem. So there's something specific about bismuth. So Helicobacter, of course, Warren and Marshall, we think about the Nobel Prize in 2005. They recognized Helicobacter as a stomach infection. It's a gram-negative bacterium. If there's a chronic infection, it can eventually lead to a form of cancer, but it will essentially lead to chronic health problems, and you get stomach bleeding, constant infections. We know roughly a lot of the targets that we have in terms of bismuth, but it knocks out some urease, knocks out some enzymes. It will also have cytoplasmic disruption, membrane disruption. So the key thing about these metals is they don't have one target. So they multi-target and they disrupt the cellular functions on various levels. Now that's fantastic because it means if the microbe develops a method to stop that, then it can find another target, and reactive oxygen species is another one. So treatment of Helicobacter is used bismuth subsalicylate, bismuth subcitrate. But the problem is that the treatment, the clarithromycin, metronidazole, and amoxicillin, the 3 antibiotics used, as you can see in this graphic, is that resistance across the world is on the rise. The one thing that resistance hasn't developed to, despite 100 years of swallowing Pepto-Bismol, is bismuth. And so it remains a core part of what you actually use in your armory of treatment for bacterial infection in the stomach. And so where the other antibiotics fail, the bismuth treatment is maintained and part of that quadruple therapy. So we went away and we've made millions of bismuth compounds, carboxylates mainly. This was the bismuth derivative of aspirin that we were interested in terms of low dose. And we can see that it's got some activity against Helicobacter. In fact, it's better than bismuth subsalicylate or bismuth citrate in treatment. So 6.25 micrograms per mil is a microbiologist's way of determining it. Chemists think in micromolar as well, but they're going to be relatively close. So the carboxylates are pretty good at knocking these things out. But we made some sulfonates, aminosulfonates. Sulfonates are common as detergents, as Andrea talked about them. And we could actually get orders of magnitude antibacterial activity just by changing the nature of the ligands themselves. So you can see in red that we are now down in nanomolar concentration activity towards these bacteria. So changing what is around the metal becomes important in terms of determining its overall biological activity. So one of the common ones you'd come across is silver. It seems to be everywhere at the moment. It's in socks, it's in band-aids, it's in bandages, it's in wound treatments, it's in baby change tables. Silver is an antimicrobial. It's been known for thousands of years, but it's not particularly effective antimicrobial. And the problem is at the moment we're starting to see some toxicity arise, we're getting resistance developing, and we're getting some environmental problems, accumulation in the environment with silver. So are there alternatives to silver that we can think of that are going to be happening? So yeah, we'll try and look at bismuth. So I'll just give you this as a little case, and then I'll come to the end of what I'm going to do. We looked at a series of compounds that are made from what's called phosphonates. You can see them here, very similar to phosphates, which you'd be very familiar with. And we made a whole series of compounds and looked at their activity against a whole range of bacteria. They're not very soluble, so I would call them insoluble, but they've got solubility of about 1 gram in about 16 litres of water. So essentially very insoluble, poorly soluble materials that don't leach out, don't provide bismuth very well. We tested these against MRSA, VRA, Staph aureus, and E. coli, and you can see that they've got activity against. This is a stab test. This is just the material itself. So we know that this form of the compound, which has an organometallic because it's got a ring just here, so it's got a bismuth carbon bond in it, is very active towards bacteria. What happens if you make what we call the tris-phosphonate or bismuth compound? So you can see now that it's got 3, the organometallic bond, so the bismuth carbon bond has disappeared. So we put 3 phosphonate ligands on instead of 2, and we get no antibacterial activity. And it's not about solubility. They're all poorly soluble. And so it's something inherent in the structure of the compound itself that allows you to tweak and change and modify the groups around the metal center that gives you that antibacterial activity. That's very difficult when it comes to molecular compounds. So as you can see, there are no zones of inhibition associated with these. So no zones attached to the MRSA itself. We then made some paper, some antimicrobial paper using nanocellulose. This is with chemical engineering. We were mainly interested in the generation of gels and also of papers themselves because we wanted to generate antimicrobial polymeric composite materials, and we put our bismuth compounds inside the paper at various loadings. What happened? Well, there's our paper. We could spray coat it because it's fairly liquid. You can just spray it down. It forms a paper sheet. It's got about, this one had about 2.5% weight bismuth in it, but we did a whole range of materials at that point. And you can see that it is the antimicrobial form of it because it's got this bismuth carbon bond here. So we put them into paper, 5, let me just draw that, 5, 10, 15, 20. You can see that the zones of inhibition, the antibacterial activity against the MRSA, VRA, E. coli, that gram-negative bacterium is very effective, but it's always more difficult with the gram-negatives to do it. So we knew the paper had antibacterial activity. We then compared it with sulfur sulfadiazine. And you can see from these plates, we can go down to 0.5 weight loading of the bismuth paper. So that's below what would normally be considered useful for an additive to a material. And we're outperforming the silver compound, the silver sulfadiazine, which is your standard silver antibacterial. So we know that we can, even against MRSA, we can get a very low loading of bismuth in about 0.1%. So we've got very good activity in these materials. The last thing I'll talk to you about is hydrogels. We wanted to generate gels for wound management infections. So just a gel that you would put on your skin if you had an infected wound and you could maybe bandage up. So we used the same components. We made a cellulosic hydrogel. And you can see from this is that round about here, about 9 micrograms per gram of compound going into the cellulosic gel, which would then be larger because it's 100% as the gel, that the bismuth is essentially non-toxic. These are towards fibroblast cells, but the silver shows a degree of toxicity. So they're outperforming bismuth in terms of safety towards mammalian cells. So that 9 micrograms per gram seems to be the best place to go. And you can see here from the live dead staining of these fibroblast cells that when you're here around about 9 micrograms per gram, when you've got lovely green cells, then they're alive, that's fantastic. And if you don't see the red cells are the dead cells, then you know that you're getting good efficacy at that point. So 9 micrograms per gram seems to be a sweet spot for that bismuth loading against the murine fibroblast cells. So what about the activity of those gels against a series of drug-resistant bacteria? So Helicobacter at 9 micrograms per gram kills A. baumannii within 4 hours. Originosa within 12 hours. Then with E. coli, we see a bit of a problem. It seems to be much more bacteriostatic. And against MRA, again, we have a bacteriostatic effect, but eventually that wears out. And against VRE, we have a bacteriostatic effect. So we don't have a bactericidal effect like we do against baumannii and against originosa, but we're definitely getting effect. A bacteriostatic effect against the other drug-resistant bacteria, which is there. So I think that shows within the sort of the ability to translate this fundamental chemistry into useful products, which should eventually find them their way onto the market. And we're using these metals to target these drug-resistant bacteria. So that's just a little sort of captured a little bit of what we do in the lab and what our students do. The last thing I want to highlight is that we got a write-up, which was good, in Chemistry World in July 2021, when we had a big article on super metals versus the superbugs. The thing is that we need to win this battle. And I'll finish it there. Thanks, Andrea.
Thank you very much, Phil. I think that we will just move straight into Rico's presentation. So over to you, Rico. Thank you.
Thank you so much, Andrea. I'm going to assume everyone can hopefully see and hear me, and I'm just going to minimize us so I don't have to look at myself because we don't want to do that. So we've heard from Phil how some absolutely cutting edge chemistry, brand new off the shop floor, can very selectively kill specific microbes without harming the rest of the organism. I'm now going to show you how 5,000-year-old chemistry can kill absolutely everything. Sounds too good to be true, right? Well, don't worry, it is. We still need the real antimicrobials. And thank you to Kelly Clarkson for providing the subtitle for this part of the talk. You'll see why that's there in a moment. Okay, so my background is in colloid chemistry, as Andrea very kindly introduced. And colloid chemistry is the science of finely divided systems. So we're talking droplets and bubbles and particles, so making emulsions and foams and particle dispersions. And I was absolutely convinced that I would go through my entire career without having to see, think, or touch a bacterium. But recently we've got very interested in making capsules, so colloidal capsules, like these ones shown up here, where we have a shell surrounding a cargo or a core that we want to protect. And it turns out these capsules are quite like cells. And through that lens, I'm going to show you my journey to trying to understand microbes. So I'm going to show you almost no research in this presentation, but I am going to show you the potentially a new way of thinking about these systems. So let's look at some microbes. Among the very wide array of different microbial systems, probably the most commonly encountered, bacteria, viruses, and fungi. And you can see they all share this fairly similar structure, and they look like a capsule. They've got an outer shell, which is a different material than their inner core. The inner core is the precious bit. It's the goo containing the brain or the life. And they want to protect that from the outside world, but they need to protect it with a membrane or a shell that can be partially permeable. This shell needs to let things in and out in order for the organism to exist and proliferate. The exact nature of the shell differs. Bacteria, it can be quite complicated, and it's going to be decorated with a whole host of different biomolecules. With viruses, it's rather simpler in its original composition because of how viruses come about, but it will often be decorated with proteins. This is a flu virus, by the way. This is not the C virus that we can't talk about, but I will talk about it at the end. And this is a yeast cell, and it's a terribly simple cell, but it's a terribly important cell. This cell wall is a little bit different. It contains an interesting polysaccharide called chitin, but I'm not going to talk in detail about fungi. I'm going to focus on bacteria and viruses. So bacteria come in, as you've seen in Phil's part of the presentation, an extraordinary diversity of forms. Now, I am by no means an expert in this field. So again, this is me learning my way through to try and make sense of how we might be able to contribute as chemists to solving outstanding problems. Before going down this path, I didn't even know the difference between gram-negative and gram-positive bacteria. Gram-negative bacteria have 2 cell walls, separated with a bit of goo and a bit of peptidoglycan, and gram-positive bacteria only have 1 cell wall. So as Phil said, the gram-negative ones tend to be a bit harder to kill, and that's the reason they've got a stronger structure. Now, these bacteria come in all shapes and sizes, and they like to stick together, and that's going to turn out to be really important as we look further into our discussion. Viruses are quite a bit simpler, but extraordinarily varied in their structure. Now, they contain an outer shell, just like our capsules, that contains the DNA or the RNA, the bit of the virus that replicates, that wants to continue existing, wants to procreate or proliferate itself. And that membrane around, the envelope, tends to be composed of cell membrane lipids. So the cell membrane lipids, for example, from our own cells. And that's because that's where viruses steal them from. They don't make their own external armor in most cases, unlike bacteria. They pinch components from their host organisms. And I'm not actually sure what this green thing is, but I think it's some kind of robot from Mars. Now, it turns out this envelope around viruses is key to their virulence, their ability to infect. And I'm going to talk mostly about the idea of viruses infecting mammal host organisms. And I'm eventually going to lead up to COVID because that's most relevant here. Because that envelope around the virus is made of the same stuff as your cells don't really recognize quickly enough that this is a foreign object. And so the virus can merge with your own cell membrane and release its virulence factor, its DNA RNA package into your cells, and there it will replicate and be re-ejected as new virus particles. We should just very briefly talk about these cell membrane lipids. I've got a few of them here, and you can see they all have somewhat similar structures. And apologies for the elaborate chemical structures. The rest you see will be quite simple. But these lipids all have something water-loving at one end, so something water-soluble. And then at the other end, they've got something that just looks like oil, that's oily and slippery. And it turns out this oily, slippery part is essential to forming this envelope, because those oily parts want to stick together to form a film, and that continuous film around the object is what encloses your biological cells, so all the cells that make up your body, and also viruses and bacteria. It's enough to make you want to wash your hands just talking about it. So let's do that. In order to wash our hands, we're going to use some surfactants, which is simply a fancy word. It's a portmanteau word, just means surface active agent, but it's the scientific term for detergents and soaps as a sort of umbrella word. And we're going to start with the world's commonest surfactant, which is sodium dodecyl sulfate. And you can see this has a very simple structure. It's somewhat similar to our lipids. It's got an end that loves water and an end that's oil, in this case dodecane or diesel. There are obviously lots of synthetic molecules and synthetic soaps and detergents on the market that can fulfill those structural requirements. But as you can imagine, this has very similar properties to a lipid. This end wants to form an oily film and get away from water, and this end wants to be dissolved in water. It's the similarity between detergents and lipids that turns out to be crucial in the way that surfactants are able to take down bacteria. So we wash our hands, we use bar soap. Actually, very few people use bar soap anymore. Its market share is tiny these days. Most people use liquid soap. I'll come back to liquid soap in a moment. But soap was first made about 5,000 years ago by heating up tallow and wood ash, so animal fat and a base, and you end up with, in chemical terms, the hydrolysis products of triglycerides, so sodium salts of fatty acids, so sodium stearate, palmitate, myristate, etc. And they happen to be solid at room temperature, giving you bar soap. And bar soap actually works fabulously well for most purposes in terms of cleaning your hands. The vital aspect of detergents here is their two abilities conferred by the part of the hydrophobic part of the molecule wanting to get away from water. One is they like to go to interfaces, so they like to do what we call absorption. They go to interfaces and stick their tails out in the air because air is hydrophobic. And by that way, they don't have to be solvated by water, or they can form an ordered structure, which again protects those oily tails from water and shields them. And this is a process known as self-assembly. It's actually the same process that results in formation of those cell membranes and those virus envelopes. If you look at a liquid soap, so very few people use bar soap anymore, now everyone uses liquid soap, you'll almost always see these two ingredients, sodium lauryl sulfate and cocamide of propyl betaine. They're fabulous surfactants. I could talk about them all day, but I'm not going to. But they are terribly ubiquitous. Now, how exactly do detergents of this type take apart bacteria and viruses? It's the similarity of these molecules to those lipids that make up the viral envelope or the cell wall of the bacterium that are crucial here. And these cationic surfactants are particularly good at it because they also have a charge attraction. So these surfactants stick into the cell membrane in between the lipids and literally dissolve it, break it apart and solubilize the membrane. This lets out the precious goo inside the cell or inside the virus particle, thereby killing it. Fabulously simple, right? So all detergents should kill all bacteria and viruses. Brilliant, we've solved the problem. Riddle me this, and I'm sorry, this is a disgusting slide. Bacteria are slightly cleverer than that, and they've developed methods to protect themselves. And one of those methods is forming biofilms. I was cleaning the bathroom the other day, and the sink was very slow to drain. And taking it apart, I found this stuff, which is a bacterial biofilm. I know what you're thinking. You're thinking, you absolute ghoul, why are you showing me this? Well, it turns out this is the mechanism that bacteria use to protect themselves from soaps. This sink in the bathroom sees more soap than probably any other piece of hardware in the house, and yet it's growing colossal amounts of bacteria. And this is the same type of biofilm, by the way, that forms the SCOBY in kombucha, that sort of fisherman's sneeze that grows at the top and ferments. It's bacteria holding the yeast cells together and doing the fermentation process. So bacteria are smarter than that, and not all soaps will kill all bacteria. What about those fabulous antibacterial hand soaps, though? Surely those are good ones, right? They must be brilliant. They were so heavily advertised when they were first around that, you know, you sort of felt if you weren't buying these expensive antibacterial soaps, you were basically murdering your family. Turns out they're awful as well. So they contain this ingredient triclosan, which was at one stage fairly potent antimicrobial, and it still is to some extent. This is hexachlorophene. Almost all of us will have been washed as newborn babies in hexachlorophene. They washed you in this in the hospital before they let you go home. These molecules are both quite unpleasant. They're carcinogenic in large quantities, but they have a much greater and more fearsome potential, which is they essentially give bacteria a workout. So if you give bacteria tiny amounts of triclosan over a long period of time, such as they're exposed to when you wash your hands and water goes down the sink with antimicrobial soap in it, then these bacteria eventually become resistant to triclosan. Well, we don't use it as an antimicrobial in humans, so probably not such a big deal. Turns out they also become resistant to a whole bunch of antimicrobials other than triclosan at the same time, which is rather extraordinary finding, but is a very good reason why we shouldn't be using antimicrobial soaps and you should just use regular soap. There really is no advantage and there's a huge disadvantage. So where should we go from here? And now I'll just give you a couple of slides on the research bits that we're doing to try and fix some of these problems, and then I'll stop talking. So we want to do soap, but better. What is better? Well, firstly, people want liquid soaps, but they've got to be viscous because you can't have something that's like water. It doesn't stay in your hand when you're trying to use it. You know, you can't wash yourself with something that just drips between your fingers. So it's got to be somewhere between a solid and a liquid. And obviously, with all our chemistry, we'd like to do things much more sustainably. So we'd like to not use petroleum products. We'd like to not use unsustainable oil sources. We don't want to use fossil carbons. And we do want to use, ideally, things that can be sustainably or at least manufactured with minimal ecological problems. Now, if you use the word bio-inspired when you write a grant, you're much more likely to get the money. So we're going to make bio-inspired detergents. We're going to use building blocks from nature. And nature has provided a fabulous set. Not only do we have carbohydrates that are water-soluble and incredibly tailorable and tunable, we have amino acids very similarly. Water-soluble, but also can be made to be water-insoluble, otherwise we'd all dissolve, right? It's the proteins that stop us being washed away when we take a shower with detergent present that wants to dissolve your lipids. And of course, nature also creates triglycerides, which are a fabulous source of those hydrophobic tail groups to make our surfactants stick together and do what they need to do. So using this idea of sustainable chemistry, because you can't say green anymore, but sustainable is definitely another money-generating buzzword, we can make these sugar based surfactants. In this case, we've linked sugar to an alkyl tail, an oily tail with triethylene glycol, which is not very green, but it's green enough. You can make it from fermentation of corn. This was a big collaboration between a couple of industrial partners, ANSTO, the synchrotron, and us at Monash. And this surfactant works fabulously. It's a great detergent. Not only that, it forms these wonderfully viscous liquid soap formulations. The reason it does that is because it self-assembles not into little spherical, tiny ball-shaped micelles, but into long strings of spaghetti through non-covalent bonds, so they can break and reform. And this is the underlying basis for all liquid soaps. Turns out you can only prove that they've done this by taking them to the nuclear reactor at Lucas Heights in Sydney and using a very rarefied experiment to unpick exactly how they respond to energy input. But trust me, they form liquid soaps. Now, the other way to get money in your grant is to use the word smart, because everything has to be smart these days. And we really hope our chemistry is smart. We tried making some smart detergents as well that had functionalities in that were known to be antimicrobial, thinking, why has no one thought of this before? We'll solve this problem in no time. And we tested these against Staph aureus, which they inhibited quite effectively, and against E. coli. And interesting, E. coli gobbled them up and actually grew much faster in their presence. So it turns out to be quite a bit harder than we initially imagined. But we've still got a few ideas in that space, so hopefully we can make some further contributions there. The other interesting thing we can do is look at, can we interrupt biofilm formation? And in this case, we're using surfactants that can be addressed with light, so we can change their shape and their solubility by shining light on them. And in doing so, we can probe the mechanism by which biofilms form. I'm not suggesting we should have photoactivated antimicrobial detergents, but they help us to understand how and why detergents can help us to take apart these systems. All right, I promised I'd get there in the end, and I am about to finish. What about COVID? Well, COVID is a really simple virus, and it's coated with cell membrane lipids from the host and a bit of spike protein. Any soap, our soap, your soap, bar soap, liquid soap, any soap will take apart SARS-CoV-2. All you need to do is wash your hands. With that, I'm going to finish and let you ask some questions. Please direct anything bacterial to Phil, as I still don't really know which end of a microbe is which, but thank you very much for your kind attention.
Thank you very much, Rico. Okay, we have a few questions on the line here. I will perhaps start with one from the chat, which is from Edward Marshall. And I think that this is directed towards you, Phil. So if it is the bismuth carbon bond that helps to give the antimicrobial activity, why do you normally use carboxylates or now aminosulfonates that you could use as antimicrobials? Are these mechanistically different?
Yep, that's a very good question. And it's not a simple answer to this. When you normally make a compound like say a bismuth compound that's derived from a carboxylic acid, it would be easy to show it if I could draw it. The easiest thing you do is you take a carboxylic acid. You could take, for example, vinegar, acetic acid. You react it with a bismuth source and you form the bismuth acetate. So you essentially form what's called a homoleptic compound. That means that on the metal center, it has exactly the same constituents all around. The difficulty comes when you want to change all the groups which are attached to the metal center. So part of the sort of benefits that metal complexes give you is that you have a kind of Lego box where you can have a metal and then you can just add different substituents. Changing each of those substituents is going to have an effect on the chemophysical aspects of that compound itself. It's also going to have a different biological effect because the biology is dominated by transport mechanisms and a thing we call lability, which is how easy is it to take groups on and off a metal center, and by a thermodynamic stability, which is how well do the compounds stay together. When you put these compounds inside a biological environment, there's many competing groups for the metal center. So you could have citrate, you could have phosphate, you could have sugars. You've got proteins and peptides galore, which are going to bind to these complexes, and they will either strip out the metal, or you want to design your compound so it gets to the cell and it can be taken up by the cell. You can add more lipophilicity, which means, like Rico, you're making them more fat soluble so they get through the membrane, or you can make them more hydrophilic and rely upon a charge transport uptake. In the end, we're trying to mimic natural processes. What we've found is that when you have what we call heteroleptic complexes, which is a metal center with different groups attached to it, the antimicrobial activity nearly always improves. And so simply moving to a structure where you have a variety of groups which have different charges, they've got different fat or water solubility characteristics, they more easily come off the metal center or they stick like glue to the metal center, you change the biological activity. What you want to do is use that capability to increase the features that you want. And so that bismuth carbon bond is a very stable bond. Most organometallic metal compounds are not particularly stable, but for a lot of heavy metals like bismuth, it is particularly stable, and it adds lipophilicity. We think that's part of the reason why there's a better cellular uptake, but there's a lot more research needed to actually try and understand why that's the case. But we see that general pattern.
Thanks, Phil. A general question here on the chat relates to, I guess it could be directed to either of you. It relates to how you go about doing your new design. And they make the comment that some of the molecules that Phil showed in the initial slides, the organic ones, are very, very complex and lots of different functional groups and stereochemistry. And yet your molecules are, much simpler in their design. So do you think about the cost of production or potential production on a large scale when you're designing your molecules, and whether that money can be recuperated by the pharma companies?
Who do you want to take it? Rico?
We'll go Rico and then you, Phil.
Okay. Yeah.
In my world, we're always talking bulk chemicals, so we don't have the luxury of being able to, to sell a medicine at a comparatively high cost, but a required very high function purity. And so when we make detergents, they've got to be real cheap. So yeah, anything more than 2 steps is probably not going to be possible for us. So those beautiful elaborate molecules I showed you are really for research to understand mechanism, and then we try and port that same chemistry across to something we can make cheaply and at scale.
Thanks, Rico.
Yeah, the initial point on the complexity of the antibiotics, the organic antibiotics which are made, is because primarily most of them are derived from natural products. And so, when penicillin was first discovered, it was because of mold on bread that's been extracted. They know that they have some antibacterial activity. And so there is a complexity in those molecules that then set the task for the organic synthetic chemist, and of which Andrea would be classified as one of those. And there's been many decades of work gone into very elaborate, nuanced synthetic development, allows you to build those scaffolds. And it's not easy chemistry, it's very challenging. Part of the reason why the pharma companies divested, let's say, in that area is because of the amount of effort that's required to design, build, test those molecules. And if you end up with one that doesn't work, there's a lot of effort gone into that. And it's a real problem if evolution is outrunning you before you can actually make the compounds which are going to be done. When we make our organometallic compounds, we look for much simpler building blocks. We generally target metals which have some function. So we look at 3+ metals such as gallium and bismuth and indium, stable metals for anti-cancer compounds like ruthenium, for imaging like rhenium. We know they're very stable. And the bismuth and gallium follow iron around the body. So what we're doing is we're creating a Trojan horse. We're essentially following something that mimics iron. If the bacteria takes those metals inside, and we just need a simple molecule to allow it to do that, then where iron is supposed to be, these metals go and disrupt the function. And so it can be a very effective approach. The components that we put around the metal, we generally use ones which are already have been studied and are understood in terms of their cytotoxicity. So we have an idea of the pharmacokinetics before we even start to build the molecules. So the aminosulfonates were based on a bigger project, and Rico talked about sulfonates. Sulfates, they'd already been studied and shown to be relatively non-toxic because they're everywhere in detergents. And so they're good groups to use. So the only thing you worry about is the effect of the metal.
Okay. All right, I've got time for a couple of very rapid fire questions and answers. So firstly to Rico. Rico, have you found out what caused the E. coli to grow rather than die when treating it with the smart surfactants?
We believe it was snipping off the sugar and literally using it as fuel. That's our best guess. They just were not sensitive to the azide part, which is normally the antimicrobial part. So they weren't interested in that at all, but they could eat the sweet bit.
It's like Coke. Okay, and a question to both of you. Do you think the COVID pandemic will lead to global changes in the use of antibiotics and antimicrobial soaps and or increased investment in this area?
Rico?
I mean, we've already seen a sort of an uptake in detergent manufacture, obviously because we've just been getting through so much more of it, detergent and hand sanitizer. I'd like to think that's going to stimulate certainly businesses looking to do their work a bit more sustainably, to invest in new molecules, and I think that really is hopefully where the future lies, moving away from non-sustainable feedstocks and practices. So if that's what we get out of this, then I think that's at least one positive.
All right, thank you. All right, for Phil. The graphs that you showed of a particular compound had bactericidal effects on 2 of the bacteria, only a temporary bacteriostatic effect on the other 2. Do you think it would be possible to use a combination of compounds that could target and kill off the bacteria in 2 phases?
Yep, that's quite possible. We don't really understand at the moment the basis of the difference in the cidal versus static behavior of it, but I suspect it's got something to do with what we call the kinetics of the process, which is how labile the compounds are, how much bismuth is taken in by the bacterium and then is actually exchanging with other metals and interrupting cellular processes. And eventually the metal will become inactivated because there's a hydrolysis and there's other mechanisms that the cell can use with it, so it doesn't exist forever. I think you can definitely design the molecules differently. You can use combination therapies, and if I had more time, I would show you some of the stuff we've done on antiparasitical work, where we use a combination of known antiparasitical drugs bound to a known antiparasitical metal moiety, which has 2 different time components to it. So you can build in that activity, which would be very useful. But again, it's research and it takes money. So need that.
Okay, and one last question, if I can squeeze it in, Kim. Rico will be fast. From Winnie. Rico, I would like to hear about the prospects of using molecular tweezers, i.e. host-guest concept, as an antimicrobial agent. It was recently developed and had shown promising results in stopping biofilm elongation in biofilm-forming microbes, such as Staph aureus, which was quite interesting. Do you know anything about this area?
I think it's a really cool idea. If you can use the sort of host-guest complexation or the molecular tweezers thing to capture a key part of the biological machinery of the virulent cell, then absolutely. I think you could use it in a very smart way to interrupt a biological process and kill the cell or stop it doing what it needs to do. So yeah, it's a really cool idea. It's difficult.
Okay. Thank you, Rico, and thank you, Phil. And I'm going to hand back across to Kim. Thanks, Kim.
Thanks, Andrea. I just thought we won't be continuing on because we could for a very long time. There's a few questions there, so apologies for those of you whose questions we couldn't answer. We're just conscious of the time and for everyone involved. So I'd like to thank Andrea, Phil and Rico for their time tonight and sharing their work, and Andrea, brilliant facilitator, fantastic. We'll be sending out a copy of the recording in the next couple of days for those of you that have requested that, and also just including a feedback form, which we'd love you to complete. So thank you everyone for attending tonight. We really hope you enjoyed it. I know Michaela and myself certainly learned a lot tonight. And actually, I know probably a little bit naive of me, but when you think of chemistry, you think of chemistry, you don't actually think the knowledge that is required from the biology perspective as well. So I found that absolutely amazing. So have a lovely evening, everyone. Thank you so much for joining us, and we hope to see you at our next one. Okay, good night. Bye.